Good morning, everyone. This is Miss Torres once again recording for you guys on this. I hope that you guys use this to listen carefully in order to better understand the section. Okay, so this section is 11-2, probability and Punnett squares. You're going to write this on page 87, 80, I'm sorry, 87, skip 88, go on to 89. You're going to put your vocab worksheet on page 88. And then if you need to, you can go to 91 because we're going to put a second worksheet on 90. So 87, 89, and 91. So you're going to keep it all on the right side this time. Okay, in addition, please make sure you write down the standards. In this case, the standards are our essential questions that we're going to be answering with this section. So let's start right up. Probability. Probability is the likelihood that a particular event will occur. You think of percentage. The common word used for probability that all of us kind of use on a daily basis is actually chance. The chance of something happening, that's basically what probability means. The chance of something happening. So for example, flipping a coin, you have a 50% chance of getting tails and a 50% chance of getting heads. If you flip it twice, what is the probability that you will land heads up both times? Well, and what humans often think of is you would say, well, if I had 50, well, the first time, 50% of getting heads the first time, the second time will be another 50, so I'll for sure get it the second time. But all of us that have ever tried that, you know that it can come up two tails in a row, and what happened to that 100% probability? It doesn't mean that the math behind probability is wrong. It means that we did it wrong. So basically, what you have to do with probability is that every time you flip a coin, for instance, like the first time it's 50%, the second time it's also 50%. And instead of adding the probabilities 50-50 to get 100%, you have to multiply them. So if you multiply 1/2 times 1/2, you get a fourth. So actually, your probability is actually lower of getting two heads in a row. So the principles of probability can be used to predict the outcomes of genetic crosses. Because we understand the math behind this, we are able to figure out what's going to happen in the future as long as we have all the data. Okay, so combination of alleles. An allele is a different form of a trait. If you remember from the previous section, we talked about what an allele is. An allele is a different form of a trait or otherwise a different version of a gene. Okay, so homologous. Homologous means two of the same. The prefix homo means the same, two of the same. So two of the same alleles or types of genes for what trait? Um, An example of this would be height. So height of, let's say, a pea plant we're talking here. We could look at tall, two big T's, two capital T's, they're the same, so we would call those homozygous. Two little t's, two lowercase t's, the two smaller t's are the same, so those are also homozygous. Round with a big R, two capital R's, that would be homozygous. Two lowercase r's, those look exactly the same. Those are also homozygous. Color, blue with a big B, or I'm sorry, that's brown actually in this case. Brown would be homozygous with two capital B's, or homozygous two little b's, because those are the same. So you could see, as long as the allele you get from your mom and the allele you get from your dad, if they're the same version, we call those homozygous. So if your mom passes down the same version as your dad for that particular gene, you wind up homozygous for whatever trait that is. Heterozygous. Hetero, the prefix means different. So hetero means different. So two different alleles or versions of the same trait. So height, for example, you can inherit a dominant or a big T from one parent and a little t from the other parent. If you get two different versions, one from each parent, you wind up being heterozygous for that trait. So here's the example of heterozygous for tall, heterozygous for shape, heterozygous for color. So these are examples of what heterozygous looks like. Okay, so there's a reason why you have to do vocabulary for this section. There's a whole bunch of words that you need to memorize if you haven't already. Okay, so here's genotype and phenotype. Genotype is the genetic makeup, little prefix gene. Think about genetic makeup. Can you see your genes? I hope you understand that your genes are parts of DNA. And looking at a strawberry, when we did the strawberry lab, could you see the DNA? No, you couldn't. You actually had to squish the DNA up and then separate the DNA out using solutions. So if you're looking at, let's say, your family or your partner sitting next to you in class or anyone else, you can't see their genetic makeup. For example, the genes for height can't be seen directly. You can't see the gene. Now, phenotype, that's different. Pheno is basically, I like to kind of describe that as, oh, think pheno, think your face. Um, the phenotype is basically what you can see, the physical characteristics that someone has. So you can see phenotypes. And what you see depends on is carried in a person's genes. So if they're carrying the genes or the genotype for tall, you can see tall. You don't see the genes, you don't see their genotype, but you could see the outcome of of that genotype displayed as a characteristic in that organism. So that organism will be tall. So if you see a tall plant, they would be basically have the genes to be tall. Short, they would have the genes to be short. So genotype and phenotype, we're talking about the genes in genotype, and the phenotype, we're talking about the physical characteristics that we have. So what you can actually see, you cannot see the genotype, we can all see the phenotype. So, can two organisms have the same phenotype but different genotypes? This is something you're going to have to answer in the Quizlet question. And a hint for answering that question. Can two organisms have the same phenotype but different genotypes? Well, look at the genes for tall here. Here's my clue. You see that there's homozygous dominant, the two big T's. There's a heterozygote, the big T and the little t, or little t, little t. Think about tall. Remember, dominant covers up the recessive. So my hint here is look at the big T, little t, what would that plant look like if it had a big T and a little t? Would it look short or would it look tall? Think about that. That will lead you to the answer. Okay, so Punnett squares. We're going to start practicing how to do Punnett squares before we get the homework. So to make a Punnett square, Punnett squares are used to figure out probabilities or figuring out a chance of something happening. Now, if you worked in Vegas in one of the casinos, you have this down to a science. There's programs that you could buy. If you're an insurance broker, you also figure out the chances that someone's going to crash and then charge them accordingly. So probabilities, there's much more complicated versions of how to do this, but basically they're all derived or taken from the Punnett square. So the Punnett square is the easiest way to figure out simple probabilities, and it has been used to create many more difficult programs. But right now we're going to learn about the simple parts. Okay, so plant height. Let's say, for instance, we have the allele for tall is dominant. So it's big T capital. The allele for short is recessive, so we use the small version for short, small version of the T. So what is the phenotype? A phenotype is a physical characteristic, just to remind you. If the if the genetic makeup is big T, what would it look like? Well, in this case, it would be tall. If you inherit the genes or the alleles, big T, little t, you get the phenotype tall because the big T is dominant and it overshadows the small t. So you still get tall plant. If the plant inherits two little t's, it'll be short. So that's the only way you get short, and that's what you have to understand from this. The physical characteristics if the genotype is this. So if you're giving the genotype and you have to figure out phenotype, that's what you're being asked to do right there. Okay, well, with all this information now, let's do the cross. You will see the T's coming down. The capitals, they don't have to go first, but we like them to go first because when we do the Punnett square, I find that students get really super confused when they do the dominant or the big T second. They'll put little t and then big T. That's a giant problem because people get confused and all of a sudden start thinking that the recessive is more powerful and will overshadow the dominant. That cannot happen. It doesn't really matter where you put the capital letter, but it does matter that you understand that the dominant combined with the recessive, the dominant always wins, no matter if you write it first or in second place. So you will see the Punnett square filling out here. Pay special attention to how it gets filled out because you will be doing a lot of these. In addition, you will possible genotypes and phenotypes. That's what you always are going to have to answer from the Punnett squares. And in this Punnett square, once it's completed, you're going to find out that it's 100% tall because they're all heterozygotes. And so they're all big T, little t. Their genotype is 100% and 100% tall phenotype because it's all has at least one dominant. Okay, so genetic practice two, Punnett square to show possibilities. P shape is round, so that's dominant. The wrinkled is recessive, so we use the lowercase of the R. What is the phenotype? What is the phenotype physical characteristic if the genotype is big R? Well, we've got round. Big R, little r, that's for you to figure out. Little r, is wrinkled. So what now your question here is, what is the combination? What is the heterozygote? What is the phenotype of this heterozygote? So, Punnett square number two. Here you've got the information. Once again, fill out the Punnett square. It's your turn to figure out the Punnett square. You will see it filled out for you. Actually, the next time you won't. So pay close attention where you put the R's and where you put the lowercase and the uppercase. Make sure to fill it out. Possible genotypes. The outcome is, once you figure it out, is 50% heterozygote genotype, big R, little r, 50% little r, 50% round, and 50% wrinkled. That's what you get as phenotype. Okay, so what is the phenotype if the genotype is this? So you're gonna questions are gonna pop up on this, so make sure you answer them correctly based on what we've done. Okay, so you're going to fill out this Punnett square now, and then you're going to fill out the possible genotypes and the possible phenotypes. So good luck. I hope you've been paying attention and you get those questions right. Okay, so types of Punnett squares. Monohybrid, you're crossing one factor. Using Punnett squares, you study two alleles or one gene. An example, seed color. So if you're trying to see if dominant and recessive, you would create like a simple Punnett square like the ones that we've been doing. The yellow is dominant, the green is recessive, and then we just go through the little parts that we just did. Dihybrid cross is where you're crossing two factors. Using Punnett square, you study four alleles at the same time. Or two genes at the same time. And we're not going to spend a lot of time doing dihybrid crosses, but they are possible. You get like 16 squares out of them. We're going to talk about how to do those later. Okay, and finally, here we are, basically a simple, the Punnett squares help us figure this out, like eye color, if it were that simple. If human eye color were that simple, this would be really easy to do, figuring out if two parents with brown eyes could have children with blue eyes. So you could see right there, would that be possible if human eyes were, by pretending, we're pretending here. Human eyes are like six or seven genes. They're not just one. But would it, if this were the case, would two parents with brown eyes be able to have a child with blue eyes? Um, see if you could spot it here. Okay. This is the end of the presentation. Please make sure you retake this if you score badly the first time. Keep taking it until you get a score that will help your grade rather than not, because this is going to go into your grade. Please email me or let me know in class if you need to retake it. Any score that's lower than a 70 will be rebooted whenever I get back to my class computer. If you take it in the weekend or after school, I will reboot it in order to for you to take it again. So keep looking to see if you can repeat it until you get a good score. Have fun. Okay, bye.